Fun. Geez, we have quite a big audience there this afternoon. I know you don't have access to the microphone or to the cameras, but if you have, I'm not too sure. I'm kind of going to chat a little bit. So just let me know if you have. I'm going to open the Q&A. So if anyone has any questions through the Q&A or through the chat box, and there's something particular that you want me to talk about over the next hour, please, I'll dip in and out. Hi, everybody. I see some names there that I recognize. Hi, everybody. If you have any queries or questions over the next hour, or there's something particular, very briefly, that I can run through with you, I will try and answer them. Daniel, you have your hand up. I don't know if you can use your microphone. Do you want to try? For sure, Leisha, I can do a quick bit on photosynthesis, no problem. You keep typing them in there to the chat box and I'll see what I can do in the meantime. Okay, I'm going to do, you keep typing them in there, guys. I'm going to share my screen. Okay. It'll be difficult to do everything in the hour, but I'll certainly try. I don't know how much I can get done with you. I know lots of people have different things that they want done in the hour, but anyway, we'll talk about it. So I'm going to talk to you a little bit about the layout of the exam. And what traditionally the first question can be, etc., etc., and then we'll launch straight into it. I hope that will be of some help to you anyway. So I'm going to use a test that I've given my own fifth years as a sample summer exam for them to remind them of what their test will look like. I suspect your teacher will lay out the test just like a leaving cert paper. That's my suspicion. So if that's the case, your teacher will lay it out into 3 sections. Section A will be short questions. Okay, again, it's going to vary. You're generally very time rich. It's going to vary between teacher to teacher, but my suspicion will be that it's going to be laid out like a leaving cert paper. That means you have short questions. Short questions just means there are short answers to them. You're going to see there that traditionally the first question is going to be on the topic of food. Okay, so just remember that for yourself. So they're more than likely, hopefully that'll work for me now in a second. It's just after quitting there for a second. So more than likely, the first question will be food. Okay, and it will be probably a short question. So you're looking about all of that. So the short questions then, you're going to look at, sorry now, this has just decided to take on a mind of its own for a second. I had myself. Okay. So I've given you a short question. Again, it could be anything from all the regions to do with food, your carbohydrate. So let's talk about what it is that you need to know about with regards to food. You should have access to a document called the Syllabus and Guidelines. Again, Google them. It will tell you everything exactly that you need to know about. You could be asked about the carbohydrates. You could be asked about the proteins. You could be asked about the lipids. You could be asked about the vitamins. You could be asked about minerals. You could be asked about water. So they are the key things associated with food, and I suppose knowing the functions of food. Why do you need food? So and yes, there are experiments associated with the food chapter. There are 4 experiments. Okay, the test, what are the experiments for? The test for reducing sugar. You could be asked about the test for protein. the tests for starch, and you could be asked the test for a lipid. So traditionally on the Leaving Cert paper, guys, question 1 is on the topic of food. That's traditionally, I can't say for certain, but historically, that is the first question. You could be asked about food. Róisín, could I be asked about the food experiments within a short question? You can be asked about the experiments in any section. I know we have a section dedicated solely to the experiments, but you could be asked about the food experiments within question 1. So yes, so this is a classic sample question that I have here for my own fifth years that I have given them in the run up to their summer exam. So you could be asked about any of those different characters. So the experiment could be asked within the short question. And I've picked 4 short questions. And of course, for somebody there that wants a question done or a little bit of revision done on photosynthesis, there is, of course, look, there's a question I've given my own fifth years on the cell organelle that is indeed the chloroplast. So within section A, that means traditionally section A is going to have short questions. The first short question, as always, could be on food. You then move on to section B. Section B is dedicated to the experiments. Okay, and within my own fifth years, they're being asked about 3 particular, they'll get 3 experiment questions and they have to do 2 out of 3. Remember, I've laid this paper out in line with the Leaving Cert exam. So yes, you could be asked about the experiments. I would say to you, again, as an experienced examiner of the 2024 Leaving Cert paper, the author of that paper in 2024, he again places massive emphasis on scientific method. So scientific method can be linked with the experiments. So make sure that when you are studying your experiments, that you make reference to the scientific method. Go back over that chapter of your textbook, or if your teacher has given you notes, make sure that you look over those scientific method notes along with the experiments. Yes, you could be asked for sure, based on the Leaving Cert 2024, you absolutely could be asked to write out the method for an experiment. Okay, you absolutely could be asked to write out the method for an experiment. If any of my own students are there, hello, good afternoon to you. I sounded like Joe Duffy there. Maybe I'll take over his new job now that he's retiring. Anyway, I digress. They know that you need my favorite number in the whole world, I will tell you, is 3. So Róisín, how many method points? I know a lot of you will have done the heart dissection. Nowhere can we afford to give you all of the marks for writing out a whole method. So I always tell them 3 of everything, 3 valid points should be sufficient. My experience as an examiner, based on the marking schemes I've used, no, they don't necessarily have to be in an exact order. 3 valid points. The one thing from Leaving Cert 2024 is safety. Safety was huge in 2024 and no students were making reference to it. So in all of your experiments, please make reference at the start to include safety. I don't know anyone here. I don't know whether you have long hair or short hair. So when I'm correcting your script, I won't know who you are, what type of hair you have, where you went to school. So say I tied back my hair, I wore a lab coat. They're all valid examples for safety there with in the method for valid safety within the method experiments. And then, of course, you're going to have your long questions there where you have supplementary exam or supplementary answer paper and you write the answers within those. I keep going back, so you're going to have long questions, as you can see here. My own students, these are the classic long questions that I gave them as part of their summer exam preparation for them. Practice labeling diagrams. Practice labeling them. Practice drawing them. You are not artists, okay? This is not an art exam. This is a biology exam. So my favorite number in the whole world is 3. 3 is the magic number. There's a song, by the way. Check it out on Spotify. So Róisín, how many never, ever, ever draw a diagram without putting labels on it. So how many labels do I put on it, Róisín? I put minimum of 3. So a diagram gets 3 labels. We never, ever, ever draw a diagram without labels. If I need to know examples or adaptive features, how many, Róisín? Always 3. Again, if you can't see my screen that I'm sharing, you need to refresh your screen because according to mine there, it says that I am sharing it with you. So examples or adaptive features always give 3. What else would require 3? I'm trying to think. Oh, 3 method points. So remember again, when you are, and not only do you need to be able to write out the experiment, so you need to be able to say to the examiner why you did a particular step. So why did you add in potassium iodide and sodium hypochlorite when you are testing for the presence of a particular substance? I added them in because I'm testing for the presence of ethanol. So again, making sure you know why you conduct an experiment step. So 3 method points in an experiment, including the safety, of course. Spelling. Spelling doesn't matter to a point in terms of the spelling. What matters is with regards to spelling, it must be phonetically correct. So what do I mean by that? So for the spelling, it must be phonetically correct. So, e.g., what do the letters ATP stand for? By the way, I'm a human talking exam paper. So if I throw out a question today, it is a question that potentially could appear on your summer exam or could appear on your leaving cert. So you need to know it. So what do the letters ATP stand for? They stand for adenosine triphosphate. Another one for some of you there, I saw your request for genetics. You might have studied genetics. So what do the letters DNA stand for? So it stands for deoxyribonucleic acid. And I'm sure there are other ones. So phonically, as long as it's okay. So let's say you spelt adenosine like this. Oh gosh, what's happened there? adenosine triphosphate. Phonically, that is dreamy, so I will give you full marks for that. However, if you misspell it, so if you wrote down this, adenine triphosphate instead of adenosine triphosphate, unfortunately you will get 0 marks. Adenine does not phonically give you adenosine or adenosine. So adenine, while adenine is in adenosine, no, you will not get the marks. It's adenosine triphosphate or adenosine. If you write down deoxyribose instead of deoxyribo, no, you won't get the marks. And I was the governor of that question in 2023 when they were asked, what do the letters a DNA stand for? So no, if you write down deoxyribose nucleic acid, instead of deoxyribo, no, you won't get the marks. Yes, I am aware that deoxyribose is the sugar in DNA, but it is not what the letters a DNA stand for. So please be aware of that won't get you the marks. Again, for those of you that have studied both photosynthesis and respiration, you need to know what the letters NAD or NADP stand for. So NAD, which is the version you use in respiration, this stands for nicotinamide dinucleotide, or adenine, sorry. Adenine dinucleotide. I wouldn't tell you a bit of information that you didn't need to know about. So if they ask you, yes, it was asked in the third paper of 2023, what do the letters NAD stand for? It's in the syllabus and guidelines. So nicotinamide adenine dinucleotide, that's what NAD stands for. That's the version you use, of course, in respiration. And then we have NADP, nicotinamide adenine dinucleotide phosphate. That's what the letters NADP stand for, nicotinamide adenine dinucleotide phosphate. That's the version which is used in photosynthesis. So again, that's just demonstrating a few little tips and tricks to you. Yes, there are deficiency disorders that let's say if you've done the food chapter, there are particular deficiency disorders that you need to know about. I'm just going to pop up, I'm just going to share with you one other thing, like for deficiency disorders in the food chapter, you need to know particular deficiency disorders. You've got a copy of this document, or you can access a copy of this document via the Institute of Education website under free resources, what I'm going to share with you now. Just bear with me while I So here is a list in about 10 seconds time of all of the deficiency disorders that you would be required to know for leaving cert biology. So for example, in the food chapter. So in the food chapter, you need to know a deficiency disorder for a lack of a water-soluble vitamin and lack of a fat-soluble vitamin. My own have chosen to do, well, haven't chosen, they've been, well, unless they have something different. Again, some of you might be home ec students, so you might decide to use one of your home ec examples, but you need to know particular deficiency disorders. So I have given you a list of all of the appropriate or prescribed deficiency disorders for Leaving Cert Biology. You can access this through the free resources, as I said, under the Institute of Education website. So there they are all for you. Now, sometimes there might be a little bit of a discrepancy between my deficiency disorder and your deficiency disorder. That's okay. Check back with your teacher, please, or check back with me. Sometimes there might be a discrepancy because they do allow, in some cases, let's say, for example, in the nervous system, the one my students have all learnt is Parkinson's, but your teacher might have studied paralysis with you. You can only study Parkinson's or paralysis in the nervous system. You can study multiple sclerosis. You can study motor neuron disease. Why can't you? Because the syllabus and guidelines, the two most important documents, they dictate everything that you need to know for Leaving Cert Biology. Access them, Google syllabus and guidelines for the current Leaving Cert Biology course. They tell you it can only be Parkinson's or paralysis. My own fifth years have just finished doing the skeleton, and I told them and I showed them that according to the syllabus and guidelines, they can study arthritis or osteoporosis. So when a student came up to me and said to me, Róisín, my teacher has studied scoliosis with me, is that okay? And I said, unfortunately, it is not. So there is a list of them all for you there on that one. Now, I'm just going to, hopefully that will be of benefit to you and make you aware of it. You can watch this back if you find I'm going too rapidly with you. Now, I did see a query or a question about photosynthesis. I think people want a little bit of that. So I'm going to just quickly go through some of these. Oh gosh, I don't think I'll have time. I wouldn't be able. A genetic cross. Maybe if I might try and get through one. The nitrogen cycle. Okay, a bit on respiration. Okay, it's impossible to get through all of these. Could you run through the? Oh. Okay, great question here. I'm going to quickly go through some of the questions. Ava asked a question. Ava, what's the question I could get on water? Ava, tell me 3 functions of water. That's the question I could get on that. And the answer there, do not write down, Ava, that prevent dehydration, Ava. No. What else could I not? Sorry, I'm just going to say that, write down the 3 functions of water there just in case. So Ava asked a question. She said, what possible question could I get on water? And I'm trying to answer as many as possible. So you could get 3 functions of water. Ava, this goes back to 3 being the magic number, Ava. Always know 3 functions, always know 3 examples. Ava, do not write down that it's to prevent dehydration or it is to keep me hydrated. That word is banned in my classroom. So number 1, it's a temperature regulation. Number 2, it is a good solvent. Number 3, it is a medium for chemical reactions. You could, of course, say it's involved in chemical reactions. Number 4, you can say it's transport. There are lots more. That list is not exhaustive, but I would definitely make sure that I would know 3. 4, and the minerals, Ava, it could be anything to do with, well, you're supposed to know an example of a mineral, such as calcium, and know its function and a deficiency disorder associated with that. So again, there's 3 functions of water. Let me briefly, as I said, it's impossible. I'll try and just I know I'm just going to keep repeating myself. So let me talk to you a little bit about photosynthesis and respiration. And again, I suppose I am preparing you for your summer exam. But I am also preparing you for your leaving cert and giving you a few hot tips with regards to this. So with regards to both, let's talk to you. I'm just going to again do a very crash course with you on photosynthesis. And in my experience, this will get you full marks. So with regards to photosynthesis, I always tell my students never to go into their exam, never, ever, ever to go into their exam without knowing the balanced chemical equation for photosynthesis. So you might be writing this down. Now, if you're here and you've decided to sign up to this, I hope nobody is on their phone. You're being disrespectful to yourself and to your education by being on your phone. So photosynthesis, so it is a 6CO2 plus 6H2O, arrow, C6H12O6 plus 6O2, light, and chlorophyll. Again, yes, everything that I have written there, you need to include that in order to be able to get full marks in it. Do not go into your exam. If you leave out one number, let's say you left out that 6 there, immediately you're down to 3 out of 6 if everything else is perfect. So let's say you had that in. Let's say you forgot the O here. Everything else was perfect. 3 out of 6. Leave out one number and one letter, you are down to 0 out of 6 immediately. Again, this is coming from an examiner's point of view, a leaving cert, advising examiner, if you want me to be so exact with my title. That if you leave out any of those things that I've advised you on, you are going to immediately start dropping marks. Yes, I would insist on light and chlorophyll in it. So photosynthesis occurs in the chloroplast. Again, be able to recognize a diagram of a chloroplast. In my summer exam that I just gave them here, I'm going to have to stop looking at those questions there or it will freak me out and I'll start reading them. But in my own summer exam sample that I gave them, you can see here, there is a chloroplast. That's where photosynthesis will occur. It's the location of photosynthesis, and the type of microscope, of course, that you need is an electron microscope. Louise, if you write down an electronic microscope, 0 marks. So that's where spelling does matter. Electron and electronic are not phonically the same. So it's an electron microscope is the type of microscope that you need. So the location, the location, of course, is the chloroplast. So let's talk about it. So you're saying to yourself, answer these questions for yourself. How many stages, Róisín, are in photosynthesis? That's an easy one, Róisín, you started off with. There are 2 stages, okay? So we have stage 1 and we have stage 2. Now, there is an alternative name. What alternative name can be given to it? Stage 1 is called the light stage and stage 2 is called the dark stage. What alternative name, Róisín, can be given to the light stage? Of course, it can be called the light dependent stage. And of course, the dark stage can be called the light independent stage. What does it mean? I'm a human talking exam paper, so if I ask you a question, it's a potential question that come up on your leaving cert. What does it mean, Róisín, to be light dependent? If I write down, it depends on light. So let's say Róisín writes this down. Again, I'm giving you more tips and strategies and tricks than anything else. So if Róisín writes down the light dependent, it depends on light. How many marks is Róisín going to get? Róisín is going to get 0 marks for that. Why? Because all Róisín has done is repeated the word that's given in the question. So that won't get you any marks. There's another tip for you today. Exam tip. Are you locked in? Are you listening to me? What is meant by light dependent? We can't repeat the word that's given in the question, so we're going to replace it with needs light, requires light. So it needs light. The dark stage does not need light. Now, I know your teacher will have given you this insanely complicated diagrams or descriptions within your notes. I'm keeping it simple. This will get you full marks in a Leaving Cert exam if you were to write this out for the events of the light stage, the events of the dark stage, of course. I'm just giving you what you need to write down. So we know within the light stage, there are two events to the light stage. Sorry, I keep hitting my keyboard. We've got cyclic is pathway 1. And we have non-cyclic is pathway 2. So if you write down these things in the exam, if you're asked, I'm sure you all saw, I'm going to show you my 5th year summer exam again. I hope I have this right. I have so many different things going around in my head at the moment. Yes, I did. So if you notice here, my own 5th year's in their sample summer exam. Look what I asked them. I'm very mean to them. The biochemical process in photosynthesis can be described under 2 main headings, the light stage and the dark stage. Give a detailed account of each of these stages. And again, if any of you are familiar with doing past paper questions, this was either on a Leaving Cert or a deferred Leaving Cert paper. So they were official state papers. I've put up my own solutions to them. They've got access to solutions. I've written out solutions for them. And I'm just briefly I can show you those exact solutions that I've written for them. But this is just me rewriting what we say about in pathway 1 in cyclic. What's the feature about cyclic? In pathway 1 in cyclic, the electrons return to the chlorophyll. And in pathway 2, the electrons do not return. So remember, there's so many different questions asked that any valid piece of information that you write is going to clock you up marks straight away. So for example, some of you might write these 2 statements down and you might get 6 out of the 9 marks straight away before you even write down anything else. Give me one other piece of information and you've got full marks for describing the light stage, believe it or not. So what happens in pathway 1? Well, the only thing that gets made in cyclic is ATP. So now I'm saying you need to tell the examiner, tell your teacher, how did you make ATP? So describe to your teacher. So describe to your teacher the formation of ATP. Okay, don't just write that down, what I have there, please. You need to start telling me that ADP plus P plus energy which has come from light, which is energized or excited the electrons. Yes, I know your teacher has gone through passing from one electron transport carrier to another, but you don't need all of that, okay? This here, describing this will be fine. So that's what happens. In pathway 2, again, you would describe the formation of ATP. You would describe the formation of NADPH. And you would describe the photolysis of water. So that's what's happening now. Again, if you'll say to yourself, how do I make ATP? I make ATP by ADP plus P plus energy. Where has this energy come from? This energy has come from electrons that have been energized or excited by sunlight. They join together, they make ATP. It's exactly the same way here. How do I make a molecule of NADPH? Again, you start off with NADP+. That is a VIP taxi. That's what I tell them always. It is a taxi. What do taxis do? They transport passengers. What are the two passengers that NADP plus transports? It's a carrier of hydrogen ions and electrons. So you might never have heard of hydrogen ions, but ask yourself, have you heard of protons? Protons and hydrogen ions are exactly the same thing. Your teacher might have taught you that they are protons because your teacher is afraid you'd forget the ions part of hydrogen ions and you just write down hydrogen. And you will get 0 marks if you just write down hydrogen. So you have your NADP plus hydrogen ions plus electrons. That's how you make NADPH. And Leaving Cert exam paper has asked you that exact question. How do I make a molecule of NADPH? So we talk about NADP plus. Who gets into the back of the taxi first? The electrons. All of my chemistry students will know, biology teachers, it's even in your textbooks, that E with a line over it is the symbol for electrons. You don't need to have them balanced. We are not chemists. So if you just write down E, electrons have a negative charge. They make it go to NADP minus. Who gets into the back of the taxi then? The hydrogen ions or the protons or the H plus, and that makes NADP. Okay, and then you describe photolysis. Photolysis is the splitting of water by light into its 3 constituents. So what happens is water, it gets split by sunlight. What does it get split into? It gets split into 2 hydrogen ions, it gets split into 2 electrons, and it gets split into half an oxygen. So that in itself is describing pathway 1, describing pathway 2 of the light stage. Again, I'm going to highlight ATP. I'm going to, oh gosh, this is terrible. Anyway, I'm going to highlight NADPH and I'm going to highlight oxygen. You're going to say to yourself, why did Róisín highlight those 3 things first and foremost? Then you're going to say to yourself, why did she highlight 2 in pink and 1 in yellow? Well, she highlighted all 3 of them because they are the products of the light stage. And she highlighted ATP and NADPH because they are the 2 products of the light stage that go to the dark stage, where they are used to convert CO2 into glucose. So that's just a brief summary. So the 3 products of non-cyclic are ATP, NADPH, and half an oxygen. Why are 2 highlighted in pink? The 2 that are highlighted in pink are the going to head to the dark stage, and the oxygen is either used in respiration or it is released into the atmosphere. I'm going to, somebody said to me, I saw there, it's impossible in the time that's allotted to do the various different things that people want. I'm going to share just some resources or things or strategies or tips or tricks. So that's a little crash course just on the light stage of photosynthesis. I'm going to talk to you in a moment about respiration. So you might want to, again, if I'm going too fast, just get access to this recording and play it back, freeze it, pause it, write down anything that you want written down. I saw somebody's request there for a genetic crosses. Again, it would take us ages to go through them here now, but I'm going to share with you something. I'm just after spending 2 hours doing genetic crosses with sixth years this morning. So let me just share this template with you. So, um, and this night might not work for you. Again, as always, teachers can't learn definitions for you. That's your job. Teachers give you these definitions. Look at all of these definitions. So you need to, yes, I know I spoon feed my own students. I give them all to them. But you take ownership. Don't be waiting on anybody to spoon feed you. You go and find all of the definitions that you need to know for a particular chapter. How will I know, Róisín? What are all of the definitions that I need to go to? Easy. Go to the syllabus and guidelines. Put them into Google. And you will ask syllabus and guidelines for the current Leaving Cert biology. It will tell you everything that you need to know. So, of course, there's always definitions. These are all of the definitions associated with genetics that you can be asked about. Again, others like that and others along the way. Now, this is my personal experience with my own students. This is the feedback that I've gotten from them. Sorry, I'm just scrolling down to one that this template here. This is not, I got this question this morning, Louise, and they asked me, Róisín, is this template given to us in our Leaving Cert for a genetic cross for anyone that's doing it? And I gave them a little bit of a chuckle. This is a Róisín resource. Okay, this is not something you will get in your Leaving Cert. They will not give you this. They said they find it invaluable. It guides them. It's like a step-by-step guide through a genetic cross. So they don't get this. But my sixth year students that have just gone out, or the sixth years, yes, they're now in first year in university. What they say to themselves is they learnt this template off by heart. They photoshopped it from the page in front of them into their brains. And the first thing they did when they went into their Leaving Cert exam, they took out the answer booklet and they, as quickly as they could, they recreated that template. So when it came to the genetic cross question, straight away, they got into it. They wrote out the letters. They wrote out the phenotypes of the parents. They wrote out the genotype. So it gave them, it was like their step-by-step recipe for doing a genetic cross question. So you're more than welcome to take a screenshot or a photograph of that and use it for doing a genetic cross. So you write down the letters, you write down what the question tells you about the parents, you write down the genotypes of the parents, you write down the gametes, and then you do your progeny or your offspring. Again, within these notes, they've got worked examples. So that is what I mean. If you're wondering, what the heck, Róisín, do you mean? Could you give us an example of one that you've done? So look, there's an example of one that I've done with them. So we wrote down the letters. They were given, no, these letters I don't think were given in the question. We made up our own for ourselves, but it doesn't matter. Majority of the time, the letters are given to you. So we wrote down the letters, we wrote down the phenotypes of the parents, we wrote down their genotypes, we wrote down their gametes, and then we did the genetic cross and we did the phenotype. So I'm going to just zoom out on that. Sorry, forgive me. Where's it gone? Yes, look, I'm just zooming out on both of these questions for you. So if you want to see a sample for yourself, so these are sample questions. If you want to see how I use that template to solve a question for these, by the way, I should have stipulated these. I'm sorry, I'm just trying to plug in my laptop at the same time. These are examples of dihybrid non-linked crosses. So that's how you would use the template that I've just demonstrated to conduct a dihybrid non-linked cross. You've got the information given to you, and then you use it to help you to solve the answers to your question. For those of you, I know this is frustrating. I'm dipping in and out of multiple different things with you. Those of you that have done genetic crosses, can I offer you a piece of advice? I'm sure a lot of you are confused by, Róisín, when do I start using XX and XY? I know this is me being really sassy with you now, but a little bit of reality. Doesn't matter whether we're talking about crosses to do with plants, to do with humans, to do with anything. Remember, you only get offspring being produced when a male and a female sexually intercourse or create their gametes fuse. So every genetic cross is going to involve a male and a female. That does not mean that you always write down XX and XY. This morning I did a question with a male wolf and a female wolf, and never once did I write down XX and XY. When, Róisín, do you include XX and XY? Well, the question will clearly state. If the question does not state that it is sex-linked, or if the question does not state that they want you to draw XX and XY, I don't want to see. So many times. So today we were talking about a male wolf and a female wolf, and immediately students started drawing XX and XY. I said, no. Did the question say that it was sex linked? They all went, no, Róisín. So I told you, why are you hurting Róisín's heart by writing down XX and XY? When I told you the only time you draw XX and XY is when the question tells you it is sex linked. Or of course, they just want you to identify the gender or something. But no, that's the only time you start drawing XXs and XYs. Okay, my next thing with you then, I saw some of you wanting a little bit of help with respiration. From this now. See, how's that right? Increase it now. Any better? Okay, so respiration. I'm going to spend a few minutes on respiration, and then I think I'll just leave it open to the floor for questions after that. So respiration. Sorry, forgive me. I should have emphasized the importance. By the way, again, I know we're orientating towards your summer exam. I would like to think all of your teachers will be kind to you and put a little bit of choice on the paper for you, because there's loads of choice on your leaving cert. There is no question. Okay, so if you have done rest or genetics with your teacher and it's coming between you and your sanity, and you're like, my teacher says I'm going to fail my leaving cert if I don't know genetics. Well, your teacher is, that's not factually correct. Let me tell you, as an advising examiner, that statistically genetics is the least popular question on the paper. Only 22% of students will attempt the genetics question. Traditionally, does a genetics question come up every year? Yes, it does. Will every student attempt it? No, they won't. Between 96 and 98% of students that sit leaving cert biology will attempt the ecology question, but very few will attempt the genetics. Of those 22%, about 6% will get a H1 in it. So it's a very poorly attempted. Nothing is mandatory. What traditionally does make an appearance on the leaving cert paper every year, Róisín? Food, ecology, photosynthesis, respiration, genetics, cell division, human reproduction, plant reproduction, if any of you have done plant reproduction. Nothing is guaranteed. If you are listening to any teachers on TikTok, if you are listening to your own teacher, nothing. I've had so much misinformation given to me that I nearly doubt myself. The only thing that I can guarantee you is that it's a 3-hour biology exam. And of course, you've got your section A, your section B, and your section C. Good news for you. Helen McEntee announced in recent weeks there over the Easter break, she told you that the assessment arrangements for the class of 2025 will transfer to the class of 2026. That was good news for you, because normally the sixth years don't find out until the end of September in sixth year, which is not fair. It allows you. So the only thing I can guarantee to you is it's a 3-hour exam. And she has now, and again, Google assessment arrangements for leaving cert 2026. It's just a document. I've shared this document with my own 5th year students, so it's very easily accessible. She has told you what experiments are examinable in question 8, what experiments are examinable in question 9, and what experiments are examinable in question 10. But I can't guarantee very much more than that. So John Loughlin, he's the gentleman who writes your leaving cert biology paper. He might decide this year that he's not putting any genetics on it. He is well within his right. Now, that would be a huge, massive shock if he didn't put something on genetics on the paper, but they are the things that I can guarantee. So if you've covered those in school with your teacher, I suspect your teacher will put some of those on your summer exam. So that's why respiration is so important. So again, just like you have the equation for photosynthesis, you are not going into your leaving cert exam without knowing your respiration or your equation for respiration. Of course, you all know that respiration is a catabolic reaction. You all know that you're going to learn your definition of a catabolic reaction. The class of 2023 for their respiration question, they got 9 marks for 3 beautiful definitions: metabolism, anabolic and catabolic. You're all saying to yourself, Róisín, so what's a catabolic reaction? It's when a large molecule is broken down into small molecules or smaller molecules with the release of energy. That unit of energy is ATP. You're saying to yourself, what do the letters ATP stand for? Róisín, that's easy. It is adenosine triphosphate. Repetition and recall lead to remembering. So I'm saying these things to you, so you play it back, you hear it again. So C6H12O6 plus 6O2, arrow, 6CO2 plus 6H2O plus, now your teacher might have taught you energy. That's amazing. I get my students to learn 38 ATP. Why, Róisín, do you get them to learn 38 ATP? Because that's how many units of ATP are produced during aerobic respiration. This, by the way, is the equation for aerobic respiration. And you need to know how many units of ATP are produced during aerobic respiration. So we have 2 types of respiration. We have aerobic and we have anaerobic. So anaerobic means there's no oxygen present. And aerobic means there is oxygen present. So with anaerobic, you've got stage 1. What's stage 1 called? It's called glycolysis. You must know the location. The location is the cytosol. Don't say cytoplasm. I think some of the textbooks and some teachers used to say cytoplasm, but in recent years they got absolutely roasted if they wrote that down, because it is not. The only correct answer is cytosol. You need to know your definition of cytosol. What is your definition of cytosol? It is cytoplasm without cell organelles. So when you get to the end of stage 1 of glycolysis, of course, you know that humans will produce, we know they produce pyruvate, but after that, they go on to produce lactic acid. What do yeast cells and plant cells produce under anaerobic conditions? Yeast and plant cells, I suppose lactic acid, and of course, just forget, reference carbon dioxide. Yeast and plant cells produce ethanol and carbon dioxide. Again, I would insist my own 5th years, I'm making sure that they all know this is a big one. I've given it on my sample. My 6th years did a sample mock paper. It's been issued to a lot of people this week. And on it, I asked them all for the test for ethanol, because if you've done the fermentation experiment in school, you will know that you need to confirm the presence of ethanol. How do I test for the presence of ethanol? It is potassium iodide and sodium hypochlorite. Some of you, spelling here does matter, okay? Potassium iodine, 0 marks. It's potassium iodide. Potassium iodine, 0 marks. Potassium iodate, 0 marks. My chemistry students would say, Róisín, what if I write down the chemical formula, KiO3, is it minus or 2 minus? I don't know. And would I get full marks for that? You sure would, boys and girls. potassium iodide and sodium hypochlorite. T. Writing down hypochloride with a D, 0 marks. Some of you will say to me, Róisín, if I write down the iodoform test instead of potassium iodide and sodium hypochlorite, would I be okay? Depends on how the question is phrased, but generally you should be okay. And a positive result is it changes from colorless to yellow crystals if they are heated. So really important that you know that. The stages of aerobic respiration. So we have stage 1, of course, which is called glycolysis. Again, the location is the cytosol. We have the intermediate stage. We have stage 2. My pen has decided it doesn't want to work anymore. Stage 2 has the Krebs cycle. And it has the electron transport chain. So stage 1 of aerobic respiration is called glycolysis. Its location is the cytosol. What's your definition of cytosol? Do I need to know it? Yes, you do. I always tell my students to refer back to, is it 2005? Define cytosol to cytoplasm without the cell organelles. I always get my students to refer back to the Leaving Cert of 2023. Name the location where stage 1 occurs. It occurs in the cytosol. Again, straight out of the marking scheme. The intermediate and stage 2, they occur in the mitochondria. Again, if I pop up my 5th year sample summer exam to you, here you will see that I've taken a Leaving Cert deferred or some paper, and I've given them an image of a mitochondria and our mitochondrion. Mitochondrion is singular, mitochondria is plural. This is a biology exam. It's not a Latin exam. They're not going to penalize you for saying mitochondria instead of mitochondrion. Maybe your teacher might be stricter than me, but anyway. This question came from an exact Leaving Cert. Look, state where it occurs in the cell. So that's the cytosol. But sorry, back to this here. This is a mitochondria. You would need an electron microscope, or if you want to say the transmission electron microscope, they're not fussy about that, a transmission part of it. So you do need to be able to recognize an image of where the intermediate and where stage 2 occurs. The last thing I'm going to do with you now is I'm going to identify for you the products of each of these stages. And then I'm going to, for the last 10 minutes, I'm going to throw it out to the floor. And if there's, let's see if I can answer any additional questions there within it. So stage 1, what are the products? ATP. Why won't this work? And pyruvate. Pyruvate is a 3-carbon compound. Just be aware of that. Róisín, my teacher doesn't call it pyruvate. They call it pyruvic acid. That's absolutely fine. You don't need to know both. You learn one or the other. The examiners will accept both. That's the 2 products of glycolysis. So then you move into the intermediate stage. The intermediate stage and stage 2, they all happen in the mitochondria, or mitochondrion, whichever way you want to describe it. What are the products of the intermediate stage? CO2, acetyl, coenzyme A. My teacher, Róisín, doesn't call it the enzyme part. My teacher calls it acetyl-CoA. No big deal. I don't care. I'm old school. I call it acetyl coenzyme A. It is a 2-carbon compound. Again, reminding yourself, could I be asked, Róisín, about enzymes? You absolutely could. What is an enzyme? It is a biological catalyst. What is it made of? It is made of protein. My Krebs cycle products. What are my products of my Krebs cycle? They are CO2, ATP, and NADH. Again, describing the formation of NADH. How do I make a molecule of NADH? What's the job of NAD or NADH? It is a taxi. It is a VIP taxi. If you don't like using the word taxi, you say it is a carrier. It's a carrier of two VIP passengers, hydrogen ions and electrons. It is formed in the exact same way as I introduced you or I told you earlier. How is NADPH made? So you start off with NAD plus two electrons. Again, I can't guarantee what your teacher will award you marks for in the summer exam, but I can guarantee you what you will be awarded marks for in the Leaving Cert. And if you write this down as the formation of NADH, this will get you full marks. So the electron transport chain then, so they are the three products. Okay, they are the three products of the Krebs cycle. Róisín, what are the, I saw a question, Róisín, where they asked me, what are the three products of the electron transport chain? The three products of the electron transport chain is NAD plus, ATP, and water. So they are the three products. Again, you've learned from me. How do I make, Róisín, a molecule of ATP? ADP plus P plus energy gives me ATP. Remember, for every molecule of ATP that's made, also a molecule of water gets made, but also make sure that you know how the water is produced at the end of the electron transport chain. So I'm just going to clarify one last time with aerobic respiration. If the question asked me, you saw the question I gave my fifth years in their sample test, name stage 1 of aerobic respiration. It's called glycolysis. Where does it occur? It occurs in the cytosol. In 2005, you were asked to define cytosol, cytoplasm without cell organelles. And what are the two products of stage 1? ATP and pyruvate. I'm a human talking exam paper, so it's a question that I could ask you, or that in your Leaving Cert. How many carbons is in a molecule of pyruvate? Easy, Róisín. It is a 3-carbon compound. So there's 3 carbons. In the intermediate stage, what are the two products? CO2, acetyl coenzyme A. How many carbons, Róisín, in a molecule of acetyl coenzyme A? 2 carbons. Where does stage 2 occur? The intermediate in stage 2 occur in the mitochondria. Do I need to be able to draw? Do I need to be able to label? Do I need to be able to recognize a diagram of a mitochondria? Yes, I do. What type of a microscope do I need? I need an electron microscope. What else was I thinking about there? Oh, yes. Remember, what are you saying to yourself? If you've taken nothing else from today, but what's Róisín's magic number? What's her favorite number? It is 3. Why is 3? So if you're drawing me a diagram, you always put minimum of 3 labels on it. How many functions does somebody ask me do I need to know with regards to water? 3 functions, 3 examples, etc. So CO2, so we move on to stage 2. The Krebs cycle, the electron transport chain, both occur in the mitochondria. Carbon dioxide, ATP, NADH produced in the electron transport chain, NAD plus, ATP, and water. Again, making sure you know how all of those are formed. Now, I'm going to see if I can answer any very quick questions with you. There's somebody there, Michelle. Sorry, Michelle, I'm insulting the pronunciation, Michelle. Right, Michelle, I'm going to go through a few of these. Um, so Michelle, you said, is that a summary of the whole experiment in 3 points or just three things you did? So Michelle, it's just three things that you did. What would you have to, yes, would you have to specify what water transports, Leisha? No, no, you wouldn't, Leisha. I think that's a great question, Leisha. And Ava said the same, was it? Do you have to explain the functions of water? So just give three functions, Ava, but you don't have to explain. So if it's transport, you don't have to say what it is transporting. Nicola, where do you access the recording? That's a great question, Nicola. Um, I'd say, Nicola, this is just a suspicion, Nicola, but I'm sure you're going to get a link with a recording to this or something. If not, just contact the Institute website or the main office on Monday. I'd say you're going to get an email with this recording in it. Do I have, Amy Walsh? I sure do, gal. I sure have a list of all of the definitions, Amy, associated with ecology. And Amy, or do you know what's even better? Do you know what I'll share with you, Amy? On Monday, there's any resources we want to share, they'll be shared with you on Monday. So Amy, hold me accountable. I will share with you a Quizlet on ecology definitions for every ecology definition that you need to know, Amy. Where can you get access, Leisha, to the 6-year mock you made? This is very cliched of me to say. So, Leisha, on Monday, there's an announcement. Keep an eye on the Instagram or the Institute socials. I'm publishing it next Thursday. I think is the 15th. Anyway, keep an eye on the socials early next week. There's going to an announcement, Alicia. All you do, it's free. It's free, free, free. Next week, it's going to be you just register, you get access to it, and you'll also get access to the solutions. No, great question, May. So I think May, forgive me if I pronounced your name incorrectly. May, no, it does not matter whether you write in the past or the present tense. Bullet points, absolutely not. Yes, Leisha, great question. Leisha, do I need to know how many ATP are made at each stage of respiration? Technically, you do, Leisha. Very quickly, Leisha. In stage 1, Leisha, there is 2 ATP made. And Leisha, in stage 2 of respiration, there is 36 ATP made, Leisha. What is important? I'm going to say you're welcome, Michelle. Um, I'm going to quickly, Leisha, are you talking about what is important? Is this for Leaving Cert 2025, Leisha? What you think is important? I think is what you mean there. I do have a document, Leisha. Yes, Leisha, 2000. Okay, Leisha, this is I'm going to share this with you, darling. Okay. Now, Leisha, this is not Róisín's recommendations. Okay. This is not her tips. It is not anything like that. This is just, Leisha, things that you need to look back over the weekend before. Okay. Can we be very clear on that? So, oh no, it's not this one. I'm going to pop this up, Leisha, and it's up to you what you do with it. But it's not my tips. These are just things I think that you would want to look over. You won't have time to go back over the whole course, Leisha. So this is, I'm showing you this document. Okay. So please review these topics the weekend before the exam on the 10th of June. They are only recommendations for review as you will not be in position to revise the entire course the weekend before the exam, as you will have other subjects to prepare. The only certainty is that the exam is 3 hours long, and as long as it is contained within the syllabus and guidelines, it can be assessed. But Leisha, this is where my money, these are the things I would make sure that I went over the weekend before my Leaving Cert exam. Okay. Again, I'm going to give you 5 more seconds, and then I'm okay. I saw somebody say, how would you define acetyl coenzyme A? Great question from Bilal. Hi, Bilal. Bilal, it is Bilal. It is they describe it in the exam. It could be described as an intermediate compound, Bilal, and it could also, Bilal, be described as an a carrier molecule, Bilal. That's the two things. So it's just a compound. It's a 2-carbon compound which is formed during the intermediate stage. It can be described as an intermediate compound or a carrier molecule, Bilal. That's the best they could be referred to as. You're welcome, Leisha. Okay. I won't have time, darling, to go through the circulatory system, unfortunately. I'm sorry. How important, Leisha? Yeah, I have it as one of my 4 systems, Leisha, that I think are appearing for Leaving Cert. I could be wrong, Leisha, but it's one of my 4 systems that I have on my Leaving Cert for this year. I have the nervous system, I have the urinary system and the skin. I have the human reproduction, and I have the musculoskeletal system, Leisha. They're my 4, darling, that I am making sure that you'd have particular emphasis on. Now, I could be wrong, Leisha. Could be so long, but I definitely make sure that I'd know my urinary system. You're all very welcome. So keep me accountable there. I think it's Amy or Nicola. Amy, I will promise you I will share that Quizlet with you. You're very welcome. What do we need to know, Michelle, for enzymes? No problem, Meso. It was a pleasure. Very quickly about enzymes. Know the definition of an enzyme. It's a biological catalyst made of protein. Know what is meant by specificity with relation to enzymes. Each enzyme works on one substrate only. No examples of enzymes. So lipase, so it's linked massively to the digestive system. So no lipase, amylase, protease. Yeah, they would be particular. And of course, the active site theory. And you do have 3 enzyme experiments. Leisha, for the enzymes, um, be careful about, also for the optimum temperatures, is it best to give a range, or one temperature? It's up to you. It's up to you. So, like for example, they do accept a 5-degree range, Leisha, for the plants and for yeast enzymes or fungi enzymes. But if you're talking about a human enzyme, that range is very narrow. It's 36 to 38. Is it important to know homeostasis in plants, Louise? No. I'm not saying it's not, but I've more important things going on. You should have more important things going on in your biology than a homeostasis in plants. It wouldn't be my biggest priority. What controls, Leisha, what controls voluntary movements? No, 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 no. So Leisha, the answer there is the cerebrum. This, wherever you, again, if you're looking at marking schemes, if they are they mock marking schemes, or I think it was a mock marking scheme, was it from Examcraft 2024, Leisha? I'm thinking about, it's a mistake. The only answer is the cerebrum. Okay. What do we need to know for homeostasis? Okay, you just well, I suppose the definition with regards to homeostasis is maintaining a constant internal environment in a living organism. You need to know the definition. Can I give examples of homeostasis? They're kind of spread throughout all of your notes. Osmoregulation is an example of homeostasis. Excretion is an example of homeostasis. Temperature regulation is an example of homeostasis. they're all keeping the pH constant is an example of homeostasis. So you're linking it to the breathing system, to the excretory systems. So making sure you know homeostasis, the definition. Do I know my definition of excretion? Do I know my definition of why is temperature regulation so important so my enzymes don't become denatured? So homeostasis, maintaining a constant internal environment in a living organism, making sure what's Róisín's favorite number? Róisín's favorite number is 3. So I would know 3 examples of homeostasis. What would 3 examples of homeostasis be? It would be temperature regulation, it would be osmoregulation, it would be excretion. Is there much of a difference between gaseous exchange and homeostasis in plants and or in humans? So gaseous exchange in humans occurs at the alveoli of the lungs. I'm not too sure what this question means, but gaseous exchange occurs at the alveoli of the lungs by diffusion. Gaseous exchange occurs at the stomata or at the lenticels in a plant. So, and they all contribute to homeostasis. Briefly go over. Okay, Bilal, I'm going to put up my scientific method notes for you, Bilal. Oh, Leisha, I don't know. Sorry, Bilal. Leisha, could a mobilized enzyme experiment come up again this year? No idea. I'm not going to say yay or nay to it. I'll give you, Leisha, a bit of insider goss, okay? So somebody that has access last year to a huge amount of scripts as an examiner and as an advising examiner. My personal opinion from all of the scripts is students were very well prepared for the immobilized enzyme experiment. So as John Laughlin, or if I was John Laughlin, I don't think it would come up again this year. I don't think he would put it on the paper. He will check something else as opposed to, but again, I can't say either way, Leisha, but I just know it was extremely well answered last year. If I were John Laughlin, that would say to me, I'm going to ask something better, something different this year. Bilal, again, scientific method. Let me show you, Bilal, your scientific method. It's not complicated. It's 1 page, Bilal. And I'm going to share it with you, Bilal. I'm going to screenshot and you can screenshot it, Bilal. My scientific method notes, Bilal, are 1 page. When they pop up, Bilal. Think they're 1 page anyway, Bilal. So, of course, Bilal, you need to know all of your definitions, Bilal, associated with scientific method. Bilal, you will find all of the definitions associated with scientific method in your textbook, or your teacher will have given them to you, or you go to the syllabus and guidelines. These are mine. I've given them all to students. And Bilal, then there's only 1 page. Look, Bilal, the rest is just past paper questions. So there is my scientific method notes. So there is everything I believe that you need to know for scientific method. So that's all they can ask you about. All right. Don't know if there's any other questions. Got it. Great, Bilal. Thank you. Okay, guys, it was an absolute pleasure, and I hope you got one thing from it, anything at all. Thank you so much, everyone. You're welcome, everybody. It's been such a pleasure. I hope you got some use from it. Thank you, Ine. Bye, Michelle. Bye, Isaiah. You're so welcome. Bye, everybody. Bye, Jay Donnelly.